Hello everyone, my name's Paul Gordon. I'm a doctoral researcher here at Texas A&M University where I work in the optical biosensing lab with Dr. Gerard Cote. And my research for the past 4.5 years has been about point-of-care microscopy for malaria diagnostics and monitoring. Here to share a little bit more about my work with you, so let's dive in. So first question, for those of you who might not be familiar with malaria as a disease, is how does this work? And really, malaria is caused by parasites called Plasmodium. There are 5 species of it that can infect humans, and they're passed to humans by mosquitoes. The parasites reproduce sexually in the mosquito-borne stage. They are injected into the body when the mosquito takes a blood meal, and then go through an asexual reproduction cycle in the human bloodstream. And that's really where people get sick. That's right here where they progress through 5 significant phases of morphological maturity, going from merozoites that are small and free-floating in the bloodstream, growing into trophozoites, or early trophozoites, which are ring stages. You can see here these dark purple spots inside of the blood cells. When they consume the hemoglobin, they start to grow, consume the red blood cell, digest hemoglobin for energy, called a late trophozoite. Eventually, when it's fully consumed the red blood cell, it's a schizont that contains lots of little merozoites. Eventually, this will turn into a ruptured schizont that has now released more back into the bloodstream, and this is how the parasites continue to grow and propagate and eventually make people very sick. It's difficult to diagnose malaria based only on symptoms because fever is very common, and not just to malaria, but with a lot of different things. Different studies will give different numbers, but one study I found said that around half of all the people who were admitted for treatment for malaria actually didn't have the disease. They were falsely admitted, so they assumed they had malaria based on their symptoms, but in the end they did not. It causes a lot of issues not being able to diagnose people properly outside of a hospital or clinic because it reduces the availability of the drugs that are needed. It increases the likelihood of drug resistance being given if it's given to people ineffectually or without proper control, and generally just as a poor standard of care. Diagnostics and monitoring efforts both contain 3 primary components that you can think of. The first is confirming, do you need treatment? It's really just saying, is the disease present? The second is measuring parasitemia, and that's what a lot of my research focused on. Parasitemia is measurement of how many parasites are in the bloodstream, and it's answering kind of how urgently do you need treatment? Are you complicated? Do you need intravenous drugs? Or are you uncomplicated? We can just give you some oral medications and you'll probably be okay. And the last deals with identifying the species of infection. Different species have different drugs that work better sometimes. And monitoring uses all of these things to answer higher level questions about where outbreaks occurring, how do we need to allocate resources, what needs to happen to kind of track the global spread of the disease. And my research fits into a bit of a niche, bringing lab-based technology outside of the clinic. So currently the point of care technology, or the low resource accessible tech, is what's known as a rapid diagnostic test, or RDT as they're called. And it'll give you a binary yes or no confirmation based on whether there are proteins that are characteristic of the parasites in the bloodstream. You put a drop of blood on this strip, it wicks along with a buffer, and it'll show some lines if you're positive for malaria, kind of like a pregnancy test or something along those lines. But it doesn't tell you how many parasites you have. It doesn't give you parasitemia. Some of the good ones will tell you which species you have. Lab-based microscopy, is the common thing. It's been around for a long time. You take a drop of blood, you make a smear out of it, you stain it, you put it under a benchtop microscope, you look at it, someone sits there and counts cells and parasites. It takes a while, it takes a lot of training and a brick and mortar infrastructure, but it gives you all the information. Nucleic acid tests, PCR, are used, but even less accessible. So the need is to find something that can provide at a minimum the parasitemia information, but be accessible at the point of care, which is what my technology aims to do. As I mentioned earlier, parasitemia is really useful because, especially for the most common species of infection, or falciparum, that can be really deadly, it can progress very rapidly from a non-complicated to a complicated state. So being able to measure someone's infection severity before they develop severe symptoms, it could be really important. And we've talked to people in the field, and they've mentioned to us that, hey, this would be really useful. If we had this, we would probably use it in addition to RDTs. So screen with RDTs and then follow up as needed with a parasitemia measurement. The other hypothesis that I've had throughout my research is that measuring parasitemia will give a really important data point for monitoring the spread of drug resistance. About 50 years ago, there was this miracle drug, everyone thought, called chloroquine, which people may be familiar with. Last year, some people thought it was a COVID miracle drug as well. But this chloroquine was going to really knock malaria out, and it did. But as it got used across the board, parasites started to develop resistances, started out in Southeast Asia, South America, started to spread, and over a couple of decades, almost all parasites, or the majority of parasites, were resistant to the drug. And while we have better drugs today, the scary thing in some ways is that the same trend is starting to repeat itself. There are hotspots of drug resistances built up in Southeast Asia and South America. But by measuring parasitemia over time, it's possible to measure the rate at which the parasites are cleared from the bloodstream. And this clearance rate is indicative of the level of drug resistance that the parasites can have. So once again, being able to have an easy way to monitor parasitemia could allow you to collect serial measurements and tell from a big picture monitoring standpoint, are these parasites drug resistant? And if so, then we can allocate resources to that specific area to prevent this population of parasites from spreading any further. The design methodology is to use a multimodal approach to leverage the strength of different imaging modalities so that you can overcome cost and size limitations in a microscope. Bright field microscopy, fluorescence microscopy are the two primary ones. Bright field for assessing red blood cell margins and things like that. Fluorescence microscopy for identifying wherever DNA is present in the bloodstream. So that's white blood cells and parasites. I also did some work initially with cross-polarized microscopy, but ended up discarding that because it didn't provide as much utility and it wasn't working as well. So to put this microscope together in the end, it's a bimodal system. So we laid out the optics, wrapped it in some 3D printed enclosures, put control systems on top of it, housed it in an extruded aluminum case that I built. You put a shroud on it to keep out the dust and dirt and light and everything like that. And then to use it, it's a clamshell. It's got a lid that opens up. You put a sample right in the middle. That can be either a cartridge that we've developed in our lab or a glass smear. If it's a cartridge, you pipette the cells into it, and then you can close this lid. These two stepper motors you see, one controls sample focus and then x-axis translation. And here's what the images look like. On the left is from the benchtop, the right is the portable. This is the same field of view, so if you compare, you can see the same cells in each. Obviously, there are some aberrations present in the portable system, but that's to be expected because it's using much lower cost optical components. Fluorescence imaging mode shows the presence of parasites in white blood cells. As I mentioned earlier, these bright spots are places where a fluorescence dye has bound to the DNA that's present not naturally in the bloodstream, wherever a foreign body is that contains DNA, or white blood cells, which also contain DNA. And you'll notice that there is better contrast on the benchtop microscope, but there's still the ability to discriminate where these parasites are. However, there's some limitations implied based on the reduced clarity and the spread of the light that shows up, not as crisp or clear of margins. And here's what an image, when you put those together in a composite, might look like. This bright spot is a white blood cell, and then you can see the small parasites inside of the red blood cells. Another couple of pictures just so you can look at them and see what they look like. And so there's some variance in what they look like, but generally speaking, these are the similar trends that are showing up. So the goal that, or what needs to happen is there needs to be an accurate way to analyze these images, these composite images, to identify what are parasites and what are white blood cells and what aren't parasites. And there's layers of specificity that would be really nice to have in terms of measuring how many of which stage of maturity the parasites are, also being able to tell whether they're intracellular or extracellular. A lot of things that could get really specific, but fundamentally the best and most important thing is measuring the ratio of parasites to red blood cells. And in order to do that, you have to be able to take every fluorescent object and identify, is this a parasite, is this a false positive, or is this a white blood cell? Not having these fancy neural networks or anything that had been developed to do my image classification, I did a lot of it by developing modifications to standard object segmentation software that's available, Fiji Image J, if anybody's familiar with that. I won't go into too many details here, but at the end of the day, I was able to kind of separate out here based on a bimodal distribution in the portable system that's kind of separates out what I believe to be parasites not based on intensity, size, and shape, although it's not a perfect system. These are scatter plots showing the blue were the raw features in terms of area, size, and intensity, and the orange are after classification. So a lot of thresholding that I did to do my initial classifications. But with that simplistic approach, these are the results. The slope of nearly 1 here, this is benchtop parasitemia measurements across the X, portable parasitemia measurements up the Y, and so seeing agreement of those would look like a line with slope equal to 1. R-squared value of about 0.94 and slope very, very close to 1 is something that I'm very pleased with, very happy about. So it's really encouraging. However, there's a little bit of a baseline bias. This y-intercept is not at 0. There's some measure of overestimation or false positive bias in the portable system in the current image analysis methodology, and there's room for improvement on the back end in terms of data processing here. So that's an overview of my work. I hope that made sense, and I hope you all find the data sets to be useful in terms of learning how to use these image processing techniques that you're going to be working on. I'll go ahead and mention, it's worth noting that the images I believe you're working with are actually from the benchtop system, not the portable system. The benchtop system we thought would be easier to work with, at least initially, because they're higher definition, so to speak. So certainly can move forward in terms of providing portable images if that becomes something that makes a lot of sense. So anyways, it's good to speak with you in a sense. Hope you got something out of this, and I look forward to seeing where things go. Good luck.